Let us now move on to genetic code. We have seen that for each of the protein to be synthesized in a cell, there is a specific code in the DNA molecule. That code is copied by the mRNA, and with the help of tRNA and rRNA, protein is synthesized in the cell, in the ribosomes. The entire mechanism depends on what is known as the genetic code. Genetic code, the form in which the message is present in the nucleic acids. In the DNA, there are 4 nitrogen bases: adenine, guanine, cytosine, and thymine. In a cell, whether it is a bacterial cell or a plant cell or an animal cell or a human cell, there are 20 amino acids involved in protein synthesis. The same 20 amino acids are arranged in various permutation combinations to form the required protein molecule. For example, in the bacteria E. coli, according to an estimate, about 3,000 different types of proteins are required for its normal functioning. Suppose you take the example of one of the most active liver cell in the human body, liver cell. According to an estimate, it requires about 3 million types of proteins. Why does a cell require such a large number and variety of proteins? It is because more than 90% of these proteins will be functioning as enzymes. So, using the 20 amino acids, a bacterial cell can manufacture 3,000 varieties of proteins, and using the same 20 amino acids, a human liver cell can manufacture 3 million types of protein molecules. How is it possible? In what form the message is conveyed to the cell? There is no comparison to the number of nitrogen bases, the number of amino acids involved in protein synthesis, and the number of protein molecules to be synthesized. If you think of each nitrogen base representing 1 amino acid, only 4 amino acids can be coded. So that possibility cannot be thought of. Suppose we think of a combination of 2 successive nitrogen bases coding for 1 amino acid, for example AA, AG, AC, AT, and so on. 16 combinations are possible, but the number of amino acids are 20. even that does not meet the requirement. So, thinking on these lines, a biologist by name Gamow in 1957 suggested the possibility of a combination of three successive nitrogen bases, a combination of three successive nitrogen bases representing or coding for an amino acid. So if it is a combination of three successive nitrogen bases, 4 into 4 into 4, 64 combinations are possible. 64 combinations are possible. Taking clue from this idea, three scientists, Corana, Nirenberg and Mathy, Corana, Nirenberg and Mathy worked with E. coli bacteria and found that the triplet code combination is the mechanism working in the cells for protein synthesis, and this triplet code combination in the mRNA molecule to represent each of the amino acids. Therefore, it is described as the triplet codon. So they were able to decipher the genetic code. This team of three scientists deciphered the genetic code, which helped in understanding the entire mechanism of protein synthesis and the way by which gene expression takes place. We shall see what are these 64 combinations and what are the amino acids coded by these 64 combinations.